BGA24, BGA24. My name is Graham. I'm from the Tree of Life Core Lab. I'm a senior research assistant in the team. And today we're just going to do a follow-up session to our protocol session, which happened on Monday. Let me just actually share my screen so everyone can. Great. Can everyone see that okay? Yeah, brilliant. Thanks, David. Yes, so overview of the session today. So on Monday, I briefly talked about our DNA decision-making process. I'll go into slightly more depth on that this morning and have a bit of a recap for everyone. And then we'll be doing together an interactive DNA decision-making exercise. So we have a piece of software called Mentimeter, which I'll ask those who want to participate to log into, and then you'll be able to sort of vote on various femto profiles, the yields of samples that we've extracted, and see how you would progress a sample down our pipeline. And then we're able to sort of see the results, perhaps discuss things if people have questions about why we would make a certain decision. Yeah, so we kind of sort of leave it open-ended and sort of see where it takes us. And then once we finish that, we'll open the floor to anyone who has any questions about any of our protocols that we showed on Monday, or if anyone has any difficult samples they're working with, sample types, things they've been struggling with, want advice on, yeah, you can put that to myself and my team, and then we'll sort of try our best to give some advice, suggestions of things that we could try. But yeah, so let's crack on the rest of the session. So our decision-making pipeline has two decision points. The first is post-DNA extraction, and the second is post-DNA fragmentation. With both decision points assessing DNA yield, quality, and species genome size. So on the left here, we have an example of a DNA extraction with a nice high molecular weight peak DNA. And then on the right, we have so DNA that's been fragmented into a good looking peak for PacBio hi-fi sequencing. So starting off with DNA extractions, we have sort of grouped our profiles into various descriptions, which we use to describe a DNA profile. And on the left, we have a list of we class as good profiles, starting with the top with a nice high molecular weight band, and then sort of working our way down and as they get sort of slightly less good. And then on the right, we have what we class as a bad profiles. And so whenever we're making our decisions, anything that has a good profile, we generally would consider to move forward into the DNA fragmentation stage. If a sample has a good enough yield, while those in the bad profile, we would fail outright, generally because the quality is too bad to proceed to PacBio library prep. So irrespective of the yield. So post-DNA extractions, we can either send our extracts into our PacBio standard library prep, where we're looking at DNA that has a yield of a minimum 500 nanograms of DNA and falls under one of the good profiles. I haven't sort of asterisks next to the 500 nanograms here, because this is probably would only pass something with that has a low yield of 500 nanograms if we have a nice high molecular weight band of DNA. Anything else would probably lose DNA during the fragmentation process, and that would fall under the 400 nanograms of DNA required for PacBio library sequencing. So anything sort of shorter than a high molecular weight band extraction, we would generally probably fail that extraction and re-extract. And then our second mode of DNA fragmentation is PacBio ultra-low input. And here we're looking at extraction yields of between 500 and as low as 30 nanograms of DNA. And that also falls under the good profile. Here, genome size is quite important for us. So anything below 1 gigabase of genome would be able to go through for ultra low input. Anything bigger than 1 gigabase generally would not proceed with the ultra low input protocol. And so, yeah, so I've given you sort of a list of the profiles and just a quick view of what those look like. So a high molecular weight band here. So we have this nice strong peak on the gel image here. High molecular weight DNA about 165 kb, very little fragmented or low molecular weight DNA to the left of that. And then when you get we add a smear into that, we kind of get some more fragmented DNA to the left of this nice high molecular weight peak here. Still a very good profile. And then it progressively gets sort of a little bit more fragmented, the DNA, and our descriptions reflect that. So here we have DNA. We have some high molecular weight DNA, so a good chunk above the 50 kb mark. So still a lot of high molecular weight DNA for us to work with the DNA fragmentation. And then following on that, we have sort of smear with the bulk of our DNA below 50 kb. So there's still some high molecular weight DNA above the 50 kb range. And then you've got DNA falling within sort of like a hump around 20 kb. So still some good DNA to work with for DNA fragmentation. And then something which we definitely wouldn't be able to work with at all is something that just has a low molecular weight band. So all your DNA is very fragmented, very low molecular weight, and not usable for any long read sequencing. So some examples of our extraction decision-making process. So here's an example of a high molecular weight band with a smear from a coral, a yield of 1,000 nanograms, a genome size of under a gig. So this is something that we would pass for a low input PacBio library preparation. Here, another high molecular weight DNA extraction. Fortunately, the yield is quite low, so 78 nanograms. However, the genome size is also relatively small, so 0.45 gigabases. And so this is something we would pass for ultra low input fragmentation. And here we have a sponge, great yield, over 9,000 nanograms of DNA, small genome size. However, the bulk of our DNA is sitting within this low molecular weight band. So generally something that we won't be able to work with moving forward. So the class this as a fail for DNA quality DNA. Another example here, we have a white-nosed dolphin. So this is our DNA extraction on the right here. You can see generally quite fragmented DNA here. However, you can see that the fragment size kind of falls within what we're looking for PacBio hi-fi sequencing, so which is 12 to 20 kb. So what we would do in this situation is do a 0.45 spry to get rid of sort of any low molecular weight stuff here, and to tighten the peak of the fragment sizes. So to try have a direct submission for PacBio sequencing. So the yield is very high, genome size is large, so we want to try aim for the low input PacBio fragmentation instead of the ultra low input. So we would class this as a direct submission. And so an example of what the output of this after a harsh spry is we've been able to get rid of some of the low molecular weight DNA, sort of tighten the peak a little bit, and then you get sort of an average fragment size, I think it's close to 11 kb. So slightly lower than we would ideally want for low input fragmentation, but you can see the mode of our DNA is sitting around 14 kb. So something that we think will work quite well with PacBio long read sequencing. And then moving on to the DNA fragmentation decision making. This is the list of profile descriptions that we have. So a good profile, something I'll show you shortly. And then we have sort of descriptions, which is mainly that we use just to sort of get an idea of whether something will need any SPRI before submitting for fragmentation. So we have good profile here. We have sort of shoulder of shorter fragments. So that's some lower molecular weight DNA just to the left of the main mode of our fragment distribution, sort of a wide base peak. And then we have sort of high molecular weight DNA present. So this is an instance where the DNA extract didn't shear very well. And so we still have some high molecular weight DNA present. So anything above 25 kb and above, we're classed as high molecular weight DNA present. Low molecular weight DNA present, this is when we just see sort of some shrapnel of low molecular weight DNA to the left of the main peak of our sheared DNA. And then a low molecular weight peak is when we have a nice strong peak. Well, not nice, but unfortunately a disappointing low molecular weight peak to the left of our ideal fragment size DNA. And so the top three we would classify as generally good profiles for our DNA fragmentation decision-making process. So for low input or standard PacBio, we want one of these acceptable profile descriptions, a yield above 400 nanograms, and an average fragment size distribution of between 12 and 20 kb. And then for ultra low input PacBio, again, we want an acceptable profile, a yield greater than 20 nanograms, and then a fragment size distribution of 8 to 10 kb. So yeah, so it's an example of where we class as a good profile with the shoulder of shorter fragments to the left here. So the shoulder can vary in height. So sometimes it would come from the midpoint of our main peak of our DNA and sort of come down here. And then for us, that's still acceptable because during the library prep phase, the shorter stuff will be size selected out. So here we have a yield of 2000 nanograms, genome size is over 3 gigs, and our average fragment size is close to 14 kb. So something that we would definitely pass for DNA sequencing. And here we have another sponge, and again, a really good looking profile here. The mode sitting at 8.5 kb with the average fragment size of 8.8 kb. And the yield for ULI is sitting at 90. So that's a good amount of DNA that we can put forward for sequencing. You'll notice there's a sort of peak here at the sort of near the lower marker of the femto report at 206 kb, but that will be size selected away during the library prep phase. So all in all, something that would definitely pass into DNA sequencing. So that's the recap over. Let me just pull up the next part of our. Okay, so I hope that most people have access to a smartphone or obviously on computers. Everyone see that menti.com? Fine, okay. Yeah. So if everyone who wants to take part would like to either use the QR code or go to the website menti.com and enter this code, we can start with our sort of interactive session. We will be going through some of these, some femtos and yields of some extractions that we've done in the core lab, and I'll ask you to sort of make a decision on how you would proceed with the sample. So I'll give you maybe a minute or so to get set up, and then we can start. I don't know if everyone can give maybe a thumbs up once they've sort of been able to log on, then we can. So I think we'll get going. So our first DNA, so we'll start with some DNA extractions for a few slides and then move on to the DNA fragmentation, and then we can have a go. And so the first one, we have a microalgae. So this extraction was quite good, I would say. We have a yield of 2,000 nanograms, a genome size of 0.28 gigabases. We have 3 samples remaining. So, knowing what sort of samples we have remaining in our freezers is also a crucial piece of information that we need to take into account. Yeah, so at the bottom here, I've got some options that we can choose. So we can pass for low input fragmentation. We can pass for ultra low input fragmentation. We can fail the extractions. We can make a direct submission, or we can pool our DNA. So what I mean by pooling is then often when we have a DNA extract with an acceptable profile, but the yield is too low, we perform a second or even a third extraction. And as long as the quality of the DNA is good enough, we combine the DNA into one tube so that we can reach the desired level of or the desired yield that we need for DNA fragmentation. So yeah, I hope everyone's had a chance to vote. Let's have a look at the responses. Oh, has everyone voted? Yes. So we have 5 responses, pass for LI. So yes, so that's exactly what we do with a sample. As I said, the yield is very nice. We've got a nice high peak of DNA. And then the genome size, even though the genome size is relatively small, we would definitely choose LI over ULI library prep in most cases. Okay, move over to the next one. So yeah, we have a ciliate. We have a yield of 49 nanograms, a genome size of 0.2 gigabases, and we have 7 samples remaining. You can have a look at the profile here, a high molecular weight peak here. We have a little bit of a hump at the bottom here, and we have sort of some low molecular weight DNA to the left here. Okay, so let's have a look at what we've decided. So yes, pass for ULI. Yeah, really good decision there. I think you can see the yield is very low. The genome size also sort of very small, so suitable for ultra low input. I think if someone had said pool the DNA and so do a second extraction, that would also potentially be a good option. So during the fragmentation steps, we do lose some DNA, but generally not more than half the DNA will be lost during the fragmentation step. So we'd probably generally make it to that 20 nanogram minimum that is required for ultra low input. So yes, so yeah, so for this one, definitely pass for ULI. However, if you're chosen pool, because we have 7 remaining samples, we could do a second or third extraction to try get their yield up to definitely ensure that we have enough DNA to put forward for sequencing. Okay. So yeah, we have a lizard. A DNA yield is sitting at around 750 nanograms. The genome size is 1.67 gigabases, and yeah, we have lots of samples sitting in the freezer. How would you proceed with this sample? So the average fragment size, something also we take into account here. So average fragment size of the DNA is 6,513 base pairs. If there's any questions during this, please feel free to unmute yourself and ask if sort of there's any anything you'd like to ask about our decision-making process. If you have sort of any doubts about what we would have done, please do sort of feel free to join in and let us know. Results are interesting. This is what we like to see.
To be fair, the pass one was me, but that's because I couldn't see the base pair count.
Okay. Yeah. Yes, fair enough. Yeah. I don't think we'd pass it for LI, Damon. But thanks for adding some variety. Yeah. So let's see here. Okay. So in this situation, and I'd be interested to know what Amy would have done, but I would agree with the majority of everyone here and to say that this would fail. So our average fragment size is sitting at 6.5 kb, as Damon mentioned, which is slightly too short for a direct submission. So if we were to spry this down, we would, so when we do a harsh spray, we're generally losing DNA sort of lower than 10 kb. So the bulk of a DNA, which is sitting around this 2,600 kb point, would be lost, and we'd have very little DNA that is of high molecular weight to put forward for low input DNA fragmentation. So unfortunately for this, because the genome size is sitting at 1.6 gigabases, we can't do a ULI. So what we could potentially do if something was under 1 gig is to spry out or to clean up this low molecular weight DNA and be left with some high molecular weight DNA, which we could then put forward for an ultra low input fragmentation. Yeah, so unfortunately, I don't think direct submission would necessarily work. And then pooling the DNA, I mean, that's a potential, although if we have many, with many samples remaining, that's something we would definitely wouldn't rule out, because we have some high molecular weight DNA sort of sitting to the right of this main hump. And so if you were to get an extract that had sort of more shear DNA to the right of this hump, you could pull this extract with the second extract and then spry out this high molecular weight peak here. But I think as we have many samples remaining, we would probably class this as a fail and then re-extract to hopefully get some more high molecular weight, high quality DNA out the other end. Does that make sense? I hope to everyone. Oh. Okay. So another lizard that we have here, DNA yield of 2,000 nanograms, a genome size of 1.9 gigabases. However, something that I put in here, which is potentially crucial, is that we only have 30 milligrams of sample for this species. So when we do DNA extractions, we ideally put in 25 milligrams of material for metazoa extractions. However, we also need material to do high C sequencing as well as RNA extractions. So we have to sort of partition this DNA into 3 bits to try and get all 3 sequencing platforms or data from all 3 sequencing platforms. Yeah, so this is slightly trickier one, I think. Let's have a look. Very interesting. Okay, so none of you have gone for the pass LI. Maybe someone's gone for the ULI, someone's gone for fail, direct submission, and pool. Okay, so I know what happened to the sample, and we went for pass LI. So unfortunately for ULI, the genome size is too large, so we won't be able to do this for ULI. Direct submission, unfortunately, I think we have too much high molecular weight to the right to be able to do a direct submission. So direct submission would sort of get rid of this main peak here, but would leave a lot of the high molecular weight DNA over to the right. And so, yeah, that wouldn't be a suitable candidate for a direct submission approach. Pooling the DNA, again, because of our sort of limited sample availability, we wouldn't be able to do a second extraction. What I would say is that we would probably we did do a spry on this, so something we would do during our direct submission process. So we did a half spry to try, if not eliminate this peak, to lower it down so it's not so prominent in our DNA profile. So do a spry, get rid of some of the low molecular weight DNA, and then reassess the DNA quality again. And so what we did is we did that spry, lowered the main peak at 2000 kb, and then still had enough DNA. So we had quite a lot of DNA to begin with to put that sample through for our low input protocol. So yes, unfortunately, I haven't given you that option of the 0.45 spry, but I just wanted to point out that we would still eventually take the sample through for our low input protocol by doing some sort of slight modifications to our DNA extraction before going through our low input protocol. So ideally trying to get that sort of low molecular weight peak sort of shrunk a little bit, and then attempting the LI library prep. Okay, here's a really strange looking sample. So this is a plant with a yield of 400 nanograms, genome size of 0.51, lots of sample remaining. And so what we have here is a little hump around sort of below sort of 10 kb, a trough of nothing, and then sort of some really high molecular weight DNA, greater than 200 kb. Yeah, sort of some high molecular weight DNA. So what do people think about this? Very interesting. Yes. So I'm glad that no one's failed it, because I would have thought that that's something that people would have gone for. Pass LI, I I think our yield is limiting what we can do for that one. So because we only have 400 nanograms, a lot of our DNA is this low molecular weight DNA sitting under 10 kb. So during the LI process, this will be lost. Or even if you recover that, we don't want that in our DNA sequencing. Pooling is definitely an option if you wanted to increase the yield a little bit more. However, I think because the genome size is 0.5, it's a good candidate for the ULI. So the ULI would sort of shear down this high molecular weight peak, and then we'll definitely have, because we're starting with 400 nanograms, even if you lose this low molecular weight DNA under 10 kb, you would still have enough yield. So we just need 20 nanograms of DNA for ULI. You can definitely put that DNA forward for sequencing. So from what I remember, we definitely did do a ULI submission preparation for this, and if I remember correctly, I think the profile was beautiful. So even though there was this hump towards the left here, we were able to get rid of that, shear this high molecular weight DNA down into good fragment sizes for ultimate input sequencing. Okay. Another plant, really high yield, so 4.5 micrograms, lots of material remaining, and sort of a genome size of 4.5 gigabases. Average fragment size sitting at 12,000 kb. Sample availability is also not an issue, so we've got lots to play with. Here we go. So we've got one for pass, 6 for fail, and 1 for direct submission. Yeah. So again, for the direct submission, I think potentially also not necessarily feasible because of this high molecular weight towards the right of here. I think you would spry off sort of a lot of the small stuff here, but we'd still be left with this high molecular weight DNA to the right. So yeah, potentially not something that we would advise. A pass for LI, yeah, I think that's definitely a possibility. I think it's something you could, if that was your only extraction, I think that is something that you could work with. Your yield is quite high, so 4.5 nanograms, although the majority of that is sitting within this 10 kb and below block of DNA. So you'd probably lose most of that, but you'd still be left with some of that here. So if that was your only extraction that you were able to perform for that species, that is something that was definitely worth trying. However, because the bulk is sort of sitting under, your DNA sitting under 10,000 kb, yeah, I think we'd fail this and try repeat the extraction. Yeah, either try a new extraction method or repeat the same extraction. Yeah. Okay, so we have a slug here. So we have sort of a yield of 240 nanograms, genome size of 2.8 gigs, no sample remaining. Our average fragment size is sitting at roughly 60 kb. So we have 5 passes for LI, 2 fails, and 1 direct submission. So, yeah, so unfortunately, our genome size is too large for a ULI, so we can't do that. Pass for LI, yeah, I think, yeah, because the yield is only sitting at 240 nanograms, we're not going to meet that. We're already below the threshold for PacBio low input sequencing, which is 400 nanograms. So during the LI fragmentation step, we're going to lose some DNA. We might, if we're lucky, end up with 180 nanograms of DNA. And especially with a large genome, you need quite a lot of DNA to get coverage for your genome. The same with the direct submission. Even if you were to be able to submit or to share that down and do a direct submission, you'd still have not enough material, not enough DNA to get a good sequencing run. So unfortunately, yeah, we'd have to class this as a fail. Ask if the sample can be recollected for us, and so that we can try again. I think if we had sample remaining, there would be a different story, because then we could aim to pool the extracts together. So we'd do a second or maybe a third extraction for the species, hope for the same quality to come out, and then combine the DNA to get sufficient yield for us to follow through for low input PacBio sequencing. Yeah, it's a pity because it's a really good looking extraction. However, the yield is sort of limiting on what we can do with this sample. Okay, so here we have the hazel dormouse, a yield of 9,000 nanograms, genome size of 3.8 gigabytes. Sample is limiting, so we don't have a lot to play with. So that's something to take into consideration. So we have 2 for parcel I, 0 ULIs, 1 fail, 4 direct submission, and a pool. So let's have a look at this profile. So we're sitting at sort of 11,000 average fragment size. The mode is close to 10. And we have some of this low molecular weight here. Yeah. So I think this is a really good candidate for a direct submission. Slightly short on the average fragment size. However, if we sort of do a nice harsh spray. We'll get rid of this low molecular weight DNA to the left of the peak here, and tighten up the peak of our DNA sitting around this 10,000 kb mark. So yeah, this is a very, for me, this is a very good example of something we would do a direct submission for. So get rid of the low molecular weight DNA, tighten the peak, and then generally we sit sort of close to that 12 kb mark for low input DNA sequencing. Yeah, pooling is potentially an option, but I mean, we already have quite a lot of DNA here. So I don't think pooling is necessarily the right way to go, because we generally pool when our yield is very low, but our quality is quite good. So the idea for pooling is sort of to get closer to better quality as opposed to increasing the yield. So we don't need a yield of more than 1,000 nanograms, or maybe 1,500 nanograms for a genome size this big. Failing is a possibility, because I mean, some of the DNA is quite low quality here, but because we have this nice sort of strong peak close to 11 kb, I think we can try a direct submission, see how that works. If for whatever reason the profile comes out poor after a 0.45 spray, we'd then sort of fail again and then try repeat the extraction. But yeah, so that's just, I mean, for me, this is a good example of what we do for a direct submission. And then we have a fish, 7,000 nanograms, lots of material remaining. The average fragment size is sitting around 23 kilobases, genome size of 3 gigabase. Yeah, so we have 5 in the pass for LI. We have 2 fails and 2 direct submission. So for a direct submission, I think the average fragment, well, for us, I think the average fragment size is slightly too big. So when we were looking at a direct submission, we wanted the DNA extraction to already fall within that sort of 12 to 20 kb range. So this is slightly too big. And so, because it is slightly too big, that means that we have sort of sufficient DNA of high molecular weight quality that we can proceed for a low input DNA fragmentation. So that's what we would do in this situation. So we have sort of sufficient DNA, 17 kb and above. So we definitely proceed with our low input DNA fragmentation for this sample. Yeah, it's good yield. Most of this sort of smaller stuff will be lost during the process, but we'd still be left with sort of sufficient DNA to put forward for a PacBio standard library prep. Okay, so next we're moving on to DNA fragmentation, and then I sort of put at the top here whether it's for ULI or LI. So LI is our standard library prep, and then ULI is our ultra low input standard prep here. So we have 5 decisions down the bottom. So the first is submit for sequencing. The second is to fail it on a poor profile, fail on low yield, fail on short average fragment size, or fail on large average fragment size. So here we have a plant, valerian, a yield of 1,760 nanograms, genome size of 3.8, and average fragment size of 16 gigabase kilobases. Just a reminder, for LI, we're looking at a minimum of 400 nanograms for DNA sequencing, and then for ULI, we're looking at about 20 as a minimum. There's no maximum limit. So, yeah, great looking profile. Our average fragment size is sitting at, I think, 14,000 kb. So sort of within that range of 12 to 20 kb for sequencing. So yeah, definitely would submit this for sequencing. So someone's gone for large fragment size here. No, for LI, our fragment size range is between 12 and 20 kb. So yeah, so that falls nicely within that window for us. Here we have an ultra low input sample. So this is a comb jelly. The yield is 20 nanograms, the genome size 0.3, and the average fragment size 1.8. Yeah, so we have sort of everyone's responded as a fail, and that is correct. I think just, as a technicality, I think what we would, so fail low yield for ULI, this yield is fine. So that wouldn't be the decision why we would fail it. So something that sits in the sort of 20 and above is fine for ultra low input. So if the profile was great, then we would submit that for sequencing. So yeah, short average fragment size. So we're sitting at an average fragment size of 1.8. But equally, I think sort of deciding that it's a poor profile is also correct. I would lean more towards to say that it's a poor profile, because you can still have a short average fragment size that has a beautiful looking peak, but it's just too short. Whereas this one, we feel that the shearing process hasn't worked very well, in terms of there's a lot of low molecular weight DNA towards the left here of the main peak that hasn't been spried away or size selected away during our spry process. So this sort of subtle difference tells us something about what's happened during our protocol. And so this tells us that the sample hasn't sheared to the right fragment length, which is disappointing, but also our spry hasn't sort of got rid of the really low molecular weight DNA. So we can go back and sort of investigate why this might be, because in the future, if we have a sample that falls within the perfect range of ultra low input, but still carries over this low molecular weight DNA, that's a problem for sequencing going forward. So we want to ensure that sort of any low molecular weight DNA that we expect to be spried away remains, that we need to ensure that that doesn't happen in the future. So by specifying something's a poor profile, we can go back and reassess exactly what the profile looks like, and why that might have happened, so that we don't repeat that. And this might also just be a carryover from a poor DNA extraction. So in many instances with a lot of our jellyfish, the DNA extractions aren't incredible looking profiles, and there's a lot of low molecular weight DNA that remains, and even with our spry and DNA fragmentation, still sort of the low molecular weight lingers on. And so we need to try optimize how we can get rid of that low molecular weight DNA. So that doesn't impact on our sequencing success. Okay. So here we have a sample of yield of 2000 nanograms, 1.8 gigabases, and fragment size of 21.7. Yes, so we have 6 people submitting for sequencing, 7, and then 1 average fragment size too long. So this is, yeah, so this is definitely a sample that's on the borderline of being too long, I would say. I think, as I mentioned, we're looking at submitting samples between 12 and 20 kb. And so, yeah, that slightly falls out of that window. But from memory, I think we did still submit this for sequencing to see how the results came back. So this is something that you could either fail and try refragment this DNA to get a little bit shorter within that window, or submit for sequencing. So yeah, it really depends on the day when you're making a decision. So sometimes decision making can be a bit subjective, but generally I think we would submit that and see what comes out the other end sequencing wise. There's still a good amount of DNA that falls within that 12 to 20 kb range, so that'll be sequenced. Even if the longer stuff doesn't get sequenced, there's enough DNA in there for the shorter stuff to be sequenced first before the longer stuff. Here we have another low input sample, the wall lizard. I think this might be connected to that wall lizard extraction that I showed you previously. I think it was the last one in the extractions. And the yield is sitting at 1,700 nanograms. Genome size is 1.9, and the average fragment size is 7.2. If anyone's interested, we have sort of 2 sort of main peaks, one sitting at 2.6 kb and the other one sitting at 9.5. So we've got 5 fail for profiles, 2 submit for sequencing, and 2 fail short fragments. 3 submit for sequencing. So yes, so this one is yeah. So something I haven't mentioned is that post DNA fragmentation, we have something that our scientific operations team does during the library prep phase, which is called a modified spry. And it's kind of another spry step that they will add to a sample if we request it, that will get rid of sort of anything below the 10 kilobase, well, 8 to 9 kilobase mark. So this little second peak, if we request it, can be removed, size selected out during the library prep phase, and we'll be left with the higher peak sitting at 9 KB. However, I wouldn't hold it against anyone to suggest that it's a poor profile, because you look at it, and I think we commonly refer to this samples like this as bat ears. They are, yeah, when we look at the femto in the morning and we see this, we're kind of disappointed. But if you have enough DNA, so that's another thing to consider, doing a modified spry. If you have enough DNA, you need to be willing to lose about half of your DNA if you're going to do a modified spry. So for this example, you have 1,700, so you'll end up with roughly 900 nanograms of DNA, and a genome size of 1.9 gigs. Yeah, so you're kind of sitting on the sort of the borderline of having enough DNA to reach coverage if you do that mod spry, but it is something that is possible to do. And then again, we have a couple of people saying short fragment size, and you're right. For low input, our average fragment size is 7.2, and that is too short for low input sequencing. However, knowing that we can do a modified spry and to get rid of the smaller fragments of DNA, that will push our average fragment size back towards the 12 KB range that we're after for low input sequencing. So having extra bits of information that I've just shared with you, our team decided to submit this for sequencing after requesting a modified spry. Again, this was a sample where it's a species where we had a limited sample, and so this was the best effort that we could do during DNA fragmentation. And from what I remember, I think we were quite we were able to reach coverage for the species. The scaffold in 50s were slightly lower than what we're aiming for, but I think with limited sample that we had for the species, we were pretty happy that we were able to get some good sequence data out for the species. Okay, so now another example from sponge, a ULI fragmentation, a yield of close to 90 nanograms, a genome size of 0.1, and an average fragment size of 5.2. Okay. So, wow. Okay. So we have 5 people suggesting submit for sequencing, 5 short fragment size. Yeah, this is again where the subjective nature of decision making comes into play. Personally, I would fail this for a short fragment size because 5.2 is quite short on the, even for ultra low input. You can see the mode is sitting at 4.5 KB. So yeah, I think when they do size selection during library prep, a lot of this is going to be lost during the library prep stage. So that, I mean, but maybe you'll recover enough to get a good library prep. So this is another one that's kind of sitting on the lower border of the fragment size distribution for ultra low input. If you were, I think, very desperate, and this was your only extraction that you had, I think submitting for sequencing and seeing what came out is definitely an option. However, I would err on the side of caution and try and do another extraction and get some better quality DNA. I mean, I don't know if anyone who has said submitted for sequencing would like to give their opinion why they would like to try that, but yeah, for me, I would err on the short fragment size decision. One more slide there. Yeah, so here's another LI fragmentation from the palmate newt. A yield of 2,500 nanograms, genome size of almost 26 gigs, and average fragment size of 17.9 gigabases. So we have 7 people submitting for sequencing, 1 fail low yield, and 1 fail long average fragment size. So the average fragment size is fine. It sits within that 12 to 20 kb range, so I think that's not a problem. Submitting for sequencing, definitely an option. I mean, definitely the right way to go. I would say the yield is 2,542. Over low yield, there is a very strong case that there is low yield because the genome size is close to 26 gigs. And so we know that submitting 2.5 micrograms of DNA is not going to be enough to get coverage for 25 gig genome. But what we would do is we'd still submit for sequencing, but know that we still have to do, maybe 3 or 4 more extractions for the species and submit that for sequencing. We will need multiple extractions or multiple library submissions for the species in order to achieve the coverage for the species. So low yield, we wouldn't fail it because of low yield, but we are aware that you will need much more DNA to go into sequencing for us to get the coverage for this species. Yeah, I think that was the last slide. Yes, that is the last. So I'm going to stop sharing there now and open up the floor to anyone who has questions about our protocols, DNA decision making, anyone who has any problematic samples or species that they're working on that they would like to ask any questions about. Yeah, please do go ahead.
There is a question in the chat from Agnes. Could you give some details on the additional modified spray after fragmentation to remove the lower peak? How would you do it?
don't know. Caroline, do you know the protocol for that? I think it's a so this is a protocol that is done by our colleagues in PsyOps. It's I think they use a special bead recipe that they sort of made up for this protocol. I don't know the details, but we can find out and definitely share that with you.
Yeah, I don't know if you can hear me. The oh yeah. Yes, they kind of, it's a preconditioning of the beads upstream of the spry process. But yeah, as Graham says, that's a process that's done in a kind of separate division of the Sanger Institute, but we can certainly see whether we can get the protocol and share that.
You're muted, Graham.
Thanks, Dan. So we have a question from Nikki in the chat. We are struggling with sequencing DNA from samples stored in ethanol for extended periods. How do you deal with samples such as these? Does this affect your sequencing outcome? We haven't worked with many ethanol stored samples, but we are in the process of doing a R&D trial on samples stored in various preservation methods. So we are having samples stored either in ethanol, DMSO, EDTA, or RNA later for a week. They are then removed from the preservation buffer and then flash frozen without the preservation. And then we're doing DNA extractions on these, and we're also going to be submitting samples for Hi-C. So we're in the process of doing some R&D in order to remove the cold chain, or in order to see if it's possible to remove the cold chain from our process. So we don't have any sequencing data yet about these samples. But yeah, but something will be made available to everyone once that is true. But for blood, though, we do have many samples that are stored in ethanol, and we don't have problems with blood samples in ethanol. So a lot of the sort of high molecular weight DNA extraction techniques that we use are sort of designed with blood and ethanol in mind. So that's not problematic for us. Blood, but I'm not sure what type of samples that come in ethanol that you're working with, Nikki. Yeah, we've had a handful of ethanol samples in. They're not. Yeah, we don't know the answer, to be honest, with ethanol. We haven't done sort of a sort of an audit of samples that have come in ethanol, but we are working on sort of seeing if this is the case, if we can preserve them. I mean, preliminary results from extractions have looked promising for ethanol, but until we get the sequence data back, we're still uncertain if this has any effect on our sequencing yet. Renata, do you want to?
Hi, am I audible?
Yeah, I can hear you.
Yes. Hi, thank you so much for this interesting session. Nikki is my colleague.
Okay.
So we are working in South Africa, and a lot of the challenges we have, samples have been collected, and then stored in the field in ethanol, and then brought into the lab. And that can be, some samples are 10, 12, 15, 14 years old. So and those samples we do find we struggle to sequence, although we get high molecular weight, well, reasonable size molecular weight, 20 to 60 kb DNA. We build libraries, the library QCs look good, but the sequences are incredibly unpredictable. So it would be really interesting to see. We also have samples that have been in ethanol for 3 months, 6 months, and those sequence fine. It
Okay,
is,
so you think
I
so
think what
the-
concerns us is our long-term storage,
Okay.
and some of these samples are incredibly precious and can probably not be resampled, or if then it would be totally opportunistic. So for us, that is quite a big challenge, and we actually don't know how to solve it. We've tried various ways, but we haven't gotten there yet. So any feedback from your side would really be appreciated.
Yeah, we haven't, I mean, so we've worked with some ethanol preserved samples, but yeah, for 20 to 30 years, we have never had, yeah, we haven't had that experience. So it's very difficult to know where to start, really. And I'm just thinking, are these from certain taxon groups? Are there certain groups that work okay, but other groups that don't, or is it just across the board that you're having a problem?
So one of the samples we're struggling with is a mammal, but we also know it's been in ethanol for 12 years plus. And one of the other samples we're struggling with, we're not sure whether it's perhaps a taxa. It's from the shark group, and that has been stored in ethanol, but not that long. We've got beautiful extractions. All the QCs look great, but sequencing is challenging. And we have another colleague that also worked on sharks in South Africa, and they had a similar problem. So we talking with the sample submitter, trying to maybe get a fresh sample and then freezing it and see if that's gives better results. But we're not at that point. We actually have to wait for the season and the chance for that sample to actually be sampled, probably within her permitting. So we actually don't know whether it's storage or whether it's species specific or a combination of both. Have you sequenced sharks or in that?
I don't know if we've had sharks yet. Have we? Amy will know. She does our coordinates. Sorry, can I just ask quickly, what sort of sequencing are you guys doing?
Yeah. So we are sequencing oxid nanopore, so we know it's more tricky. But the one mammal sample failed also with PacBio, and the Illumina sequencing was also not great. So we know Illumina wasn't great. We know PacBio wasn't great. So perhaps it's ultimately ambitious to try oxid nanopore. But we thought we'll give it a try, and it's not.
So Amy, do you know we've done sharks? You've done sharks.
Yeah, so we've done the starry smooth hound and the basking shark. Both were sort of from frozen. And yeah, the starry smooth hound was really good quality. The basking shark was sort of okay-ish, but it was workable with what we got. And yeah, the starry smooth hound sequenced really well. I can double check the exact sort of metrics we got on PacBio, but from what I remember, it was a good one to work with.
Excellent. So maybe asking for a fresh sample that is snap frozen is the way to go.
Yeah, possibly. And I think as well, we've sent, it might be the Starry Smooth Ham we've sent for ONT as well, but we've not got the results back for that. So I'm not sure how it might sequence on ONT as well.
Okay, great. Thanks.
Okay.
Great, thanks, Renato. But yeah, I guess any follow-up questions, yeah, please feel free to get in touch with us. We're very, very happy to help. I'd like to mention that now, actually, while we have the chance, that we do hold monthly R&D meetings on our various taxon working groups. So 1 month we'll focus on chordates, 1 month we'll focus on plants, 1 month we'll focus on protists, and so we'll sort of like circle through the different taxon working groups. So if anyone on this call here is interested in joining those calls, please do get in touch and we can add you to the mailing lists. We sort of report on our sort of routine or production samples, and then also sort of bring any R&D that we're working on to these meetings, any things we're struggling with, and also encourage people to bring their expertise, their issues to those meetings as well. So if you are interested in joining some of those meetings, please let us know. Okay. Alphabet. We are struggling with building libraries ONT for some of our flowering plant samples. The 260-230 ratios are low. Do you have any advice for further optimizing Heimlich rate DNA extractions if we already incorporate sorbitol washes, RNase A, BME, and chloroform steps with the Nucleon Phytopure kit from Sativa? Yes, thanks, Elizabeth. I forward your email just from yesterday afternoon onto Ben. Ben, are you on the? Yeah, Ben's on the call. And so I guess he can give you some, maybe some, I don't know, advice on what you can do plant-wise. I think you mentioned in your email that you also were working on Protea and some succulents. I don't know if that's still, yeah, something that Ben can maybe help address.
Yes,
Yeah.
yes. Hi. The particular sample that we're struggling with is one from the Protea family. But we also have a few others that are succulents or plants that are, you get them in the wild. So they're not cultivated plants. And they grow in arid areas, some of them. So it's also not always predictable. Like we have some samples that had a very low ratio and then still produced good libraries and we could sequence them. But then others, just when you qubit the library at the end, it's either too low to read or around 2 nanograms per microliter, which is way too little for sequencing for ONT.
Yeah. So we haven't. We've only recently just received two of the two species from the Proteales order. So I can't comment, unfortunately, much on that in terms of performance. But I did review the methodologies you sent through for these plants, and yeah, they do seem quite robust. It's a shame that there's not working. I'm imagining these species. I know the yeah, the succulents and the lights can be quite troublesome. They can be troublesome outside as well. I think depending on what kind of yield you're getting out of these species, it might be worth the most simplest things we can think of is scaling back on the amount of tissue input you're using. Then notice doing a direct tissue lysis. Obviously, sorbitol washing is good for removing a lot of the cytosolic compounds, but there's a lot of components that aren't removed. Obviously, organelles, the cell walls, and the likes. So if you're extracting large amounts of material like this, such as 2 or 3 grams, you can concentrate a lot of this down, and that can impede the library making and the sequencing. So we generally, when I'm processing stuff like succulents, actually quite a similar methodology as you sent through. So we'll do the sorbitol washing But one thing for the note with a soil bottle, I would recommend doing it refrigerated on wet ice, as I found in my processing that it reduced the amount of degradation of the sample, and I find that it retains a lot of the DNA, and I do not get as much fragmentation because it likely reduces the amount of activity of the sample. But yeah, I'm normally extracting a maximum 0.5 gram generally. I'm looking at 200 milligrams, which can then be pulled later, which is then purified in similar methods that you're using. Once again, I try and stick to refrigeration throughout prior to your DNA being purified. But you can pull these extractions afterwards, and a lot of times I found, particularly one that was troublesome is something called the sour fig. It is the Carbobrotis edulis, a type of solugulate.
We've had that one too.
Yeah, it's been a massive pain, but we're finding that if you do smaller extractions and then pull later on, it helps a lot. Yeah.
Yeah, I must add that 2 to 3 grams that we start off with, that's in big 50 ml tubes, and then after the sorbitol washes, we split them up into 1.5 ml epis. So that is then aliquoted up front before we do the rest of the extraction, because we struggled with yield, and then when we upped the input material that we grind with the liquid nitrogen, it improved the yield at least. But thank you for the suggestion on the refrigerated centrifugation. I think we'll definitely try that. And I also noticed on Tuesday that you use a CI step and not just chloroform. So I think I might try that as well.
Yeah, I found the chloroform isomale alcohol is the best. Has the best resolution of the phases. It doesn't seem like using phenol, but I would recommend against using that for high molecular weight
Yes.
DNA. Yeah. Okay, good.
It's nasty.
Very nasty, and I've actually found it reduces the a lot of the outcomes. Actually, there's a lot of DNA damage.
Oh, okay.
Yeah, and the yields are lower as well. I would just completely omit it really, but it doesn't seem like you're using it.
No, luckily not.
But yeah, it's quite surprising after all of that. I didn't. Yeah, if you're the soil bottle washing and then separating it out into separate extractions after the fact, even after all that, if you're still having issues, it's not the funnest suggestion, as I personally don't enjoy doing it, but maybe looking into something like nuclear isolation might have to be the avenue. It might be quite difficult, but if after all of these cleanups, and if you try some maybe using cold soil bottle wash buffer, see if that helps. But if not, it might be something the fact that you're just getting something being carried over that's impeding the sequencing, which is an issue I find with a lot of plant species, unfortunately.
Yes, I've seen protocols when they're extracted from the king protea that they extracted from the flower petals instead of the leaves, which we've been using. But they added a lot of extra clean up steps, including some microbes, which I don't really want to go to. But yeah, I think we'll definitely try the cold centrifugation step. Thank you.
Okay. Yeah, I would. Yeah, trying alternative tissue types as well might be something that could work. Yeah, the issue with the flowers, I find it brings over a lot of sugars into the extraction. It can cause a lot of viscosity.
Yes.
But yeah, it's unfortunately, yeah, it's just the nature of plants. It's really troubleshooting for a lot of these things. But if there's any way I can help, I'm more than happy to give contact details and we can communicate about this.
Thank you. Thanks very much. Yes, we've made peace with the fact that it's always the plant, it's not us. So thank you.
Absolutely.
Does anyone else have any questions about anything that we've presented or any samples? Again, we're very happy to sort of receive any questions or queries by email. So please do get in touch if you would like to. do that.
Yeah, I can't see any further questions in the Discord or the YouTube. So should we make this last call? Then we can let the very busy lab team get on with their day.
Okay. Well, thank you everyone for joining in. I hope it was useful and beneficial for you. And yeah, it's been good fun sharing our protocols and our decision making with everyone. And yeah, all the best with your genome sequencing of whatever species you're working with or interested in. Yeah.
Hope to see you all in the next session. Bye.
Yeah.